Hello everyone, my work is titled "The effect of peripheral inflammation on cortical lesions is independent of blood-brain barrier integrity." My name is Berenice Silva from Argentina. Cortical lesions represent a hallmark of progressive form of MS. Additionally, pathological studies suggest that the inflammation and neurodegeneration remain trapped into a closed blood-brain barrier in these forms. The disruption of this barrier is linked with the recruitment of inflammatory cells to the CNS from peripheral circulation. Furthermore, animal and patient studies suggest that peripheral inflammation could exacerbate the central pathology. There are no data regarding the effect of pro-inflammatory peripheral stimuli on cortical lesions. The aim of this work is to study the cortical response of repaired inflammatory lesions trapped within a recovered blood-brain barrier after peripheral pro-inflammatory stimulation. Our group developed a focal chronic cortical MS animal model based on the injection in prefrontal cortex of the adenovirus that expressing chronically the interleukin-1 beta. Systemic inflammation was induced by the injection of the same adenovector and adenobeta-galactosidase as a control. We perform behavioral tests, immunohistochemistry, and MRI. Chronic expression of interleukin-1 beta in the cortex induces a focal chronic cortical lesion characterized by blood-brain barrier breakdown, macrophage, and neutrophil recruitment, not lymphocyte, demyelination, glial activation, neurodegeneration, and meningeal inflammation associated with cognitive impairment. At 56 days post-injection, these lesions evolve a small card composed with microglia macrophage, the blood-brain barrier was restored, and the associated behavioral changes disappear. Peripheral stimulation at this time point, 56 days post-injection when blood-brain barrier is closed, exacerbate the cortical lesions, which show the recruitment of lymphocytes, not neutrophils, demyelination, neurodegeneration, and meningeal inflammation. In addition, blood-brain barrier was disrupted after this peripheral stimulation. You can see in the slide the quantification of cortical damage in both groups. You can see in this slide that cognitive impairment and anxiety-like symptoms return in reactivated lesions, and you can see the MRI results. The reactivated lesions could not be visualized in MRI in both groups. Despite of this, the cortical lesions show demyelination, neurodegeneration, glial activation, and open blood-brain barrier. The conclusion: systemic inflammation exacerbates cortical neuroinflammation and neurodegeneration independently of blood-brain barrier integrity. Systemic inflammation disrupts the blood-brain barrier. Systemic inflammation flares up the central inflammation process and changes the status of the trapped inflammatory lesions within the closed blood-brain barrier to an adaptive inflammatory response. And systemic inflammation causes recurrence of behavioral symptoms. This study showed that the impact of systemic inflammation would help to elucidate the mechanisms involved in the evolution of these cortical lesions. Thank you very much.